Hello, thank you for joining me for this lecture on species and speciation. We're going to start by asking the question, what is a species, and talking about the different ways that biologists define species. Then we're going to talk about the process of speciation. How do new species evolve? And this requires us thinking about the mechanisms that keep species separate in the first place. So let's talk about how we define species, how species are kept separate from each other reproductively, and how new species evolve. Well, the definition of a species is really almost impossible to nail down because really there are not these very discrete separations between all species in nature. So biologists have a number of ways of potentially defining where one species ends and another begins, or whether these two organisms are the same species or not. One mechanism that we can use is just by looking at the observable physical traits of organisms. So the traits that we can see expressed on the outside of their body, we can use these in what's called the morphological species concept. And this would enable us to potentially identify different species when we don't have access to other tools. For example, if you look at these birds, this is a bird that has a specific body shape and size and coloration pattern and bill shape and size. If you compare the body shape, size, and coloration pattern to that of this bird over here, then you might say, look, they look very different from each other, so we think those are different species. And in fact, you'd be correct. That is an eastern meadowlark here on the left, and it's actually much larger than this Wilson's warbler as well, which you can't tell from this picture. So here's the Wilson's warbler on the right. But biologists, by and large, use a different way of distinguishing between species, and that's what's called the biological species concept. So the biological species concept is the most widely applied tool for determining species. And this one says that members of the same species can interbreed and produce fertile offspring. So if you have populations or groups of organisms and those individuals can't breed with individuals in another population or group of organisms, if they can't breed and produce fertile offspring potentially together, then they are not the same species. This is usually how we define a species. They can breed together and produce fertile offspring. Now, you can see some of the limitations of both of these types of species concepts. For example, there's sexual dimorphism, where males appear much differently than females, and that would really make things hard for this morphological species concept because they're the same species, but they look a lot different. And even with the biological species concept, you've got problems. For example, what if it's a species that's asexual? You can't apply the biological species concept to asexual reproducers like bacteria. So in that case, you have to look at more genetic differences and try to think, okay, where do we draw the line with these genetic differences? Or look at evolutionary relationships and try to figure out where to draw the line between species. So keeping in mind that we're defining species as groups of interbreeding sets of organisms, and they can produce fertile offspring, what are the mechanisms that prevent organisms of different species from mating with each other or from reproducing successfully? And we have two primary mechanisms for preventing reproduction, and we can divide these into two groups. We've got prezygotic barriers to reproduction. These are the ones that kick in before fertilization can occur. So pre meaning before, zygotic referring to a zygote that is formed during fertilization. These prezygotic barriers are barriers that prevent fertilization from occurring between individuals of different species. Then we also have potential post-zygotic barriers. So these are barriers that prevent successful reproduction but these come into play after fertilization has already taken place. So a zygote can form, but then something breaks down, and perhaps that zygote is not able to develop successfully, or offspring are produced, but they're not fertile. Let's look at some examples of pre-zygotic barriers. Remember, these are preventing fertilization in the first place. And our first example is called temporal isolation. Now, temporal in this context refers to timing. This one is not about temperature or about rhythm or anything like that. It's just a difference in timing. So you'd have two different species that reproduce at different times of the year, for example. We have, as an example here, a Monterey pine. And this Monterey pine, this species breeds in February, which means in February, these little male pine cones are laden with pollen. They've produced a lot of pollen. They're going to release that pollen, and the wind will blow the pollen, and the pollen will land on female pine cones of other Monterey pine species. Now, it's possible that that pollen can land on a different species of pine, but perhaps a different species of pine is not breeding at that time of year. For example, the bishop's pine breeds in April instead of February. So at the time that the Monterey pine is releasing its pollen, the Monterey pine is ready to receive pollen. It's reproductively active, whereas this other pine species, the bishop's pine, is not. And the differences in the timing of their reproduction help ensure that they are not cross-fertilizing each other and no zygotes are produced. That's a pre-zygotic barrier. Our next type of pre-zygotic barrier is habitat isolation. And this just means that the two different species do not live in the same habitat within an area. So they might live in the same geographic area. For example, they might live in southwestern California, but they don't live in the same habitats. Garter snakes are a good example of this because garter snakes, some species live primarily in water and some live on land. So if they're living in water, they're reproducing in water, they're interacting with other garter snakes who live in water, they're not going to reproduce with the species that lives on land. So the fact that they use different habitats is going to keep them separated, and they're not going to breed together. This next one is my favorite as a behavioral ecologist, and you can see here that you have behavioral isolation. And this has to do with species mating rituals largely. So remember when we talked about the sage grouse and the courtship behavior, of the sage grouse? This is the male sage grouse, and he not only is really fancy, but he's also putting on a display for the females. So he's kind of bringing his tail out, and he's moving and puffing up his chest, moving things around and making a big display and making some vocalizations. And that is attractive to the female sage grouse. It's not going to be attractive to a female of a different species. So this helps keep them behaviorally isolated. They are in tune with the reproductive rituals, with the courtship rituals of their own species only. And this is really seen easily in fireflies. In fireflies, the males attract the females by flashing. So you can have these males and they're flying around. This would happen in the eastern United States regularly. If you go to a field at the edge of a woods in Tennessee in June and you're out there at 9 p.m. and it's dark, you can see these firefly species flashing. And the males will be flying up in the air and they're flashing. And one species might have a flash pattern like flash, 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 flash. And there's a female of that species. The females hang out in the grass or the leaf litter below, and the female of that species will respond to that flash pattern. So when the male goes flash, 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 the female will go flash. And then he knows that she's down there and that she's his species, so he can fly down and they can mate together. And there could be 6 or 7 different species in the same area. Another species male is going flash, 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 flash. And the female of his species can flash in response to him. So another male might go flash, flash. I think you guys get the point here, right? The males of each different species have a different flashing pattern, and the females of their species respond to that specific pattern, and that keeps them reproductively isolated as species. Now, I will say that there are some females who are predatory. If they're a predatory species of firefly, after they've mated with their own species, they could respond to the flashes of other species males, and when that male comes down to want to mate with them, they might eat him. So, sometimes these rituals are taken advantage of, but by and large, these specific behavioral rituals help prevent mating with the different species. Okay, the next type of prezygotic barrier is mechanical isolation. And the best way to think about mechanical isolation is that the parts do not fit. So in this case, male and female of different species will not be able to mate because the male part that delivers the sperm does not fit with the female part that receives the sperm. And spiders are actually a really good example of a group that uses mechanical isolation, because what I have pictured here, you can see this is a male spider, and often the males are a lot smaller than the females. You can see that this male spider has 8 legs, 1, 2, 3, 4, 5, 6, 7, 8, 4 on each side. But you might notice up here at the front of its body, there is another pair of appendages, and these are called pedipalps. And in females and males, they're used to sense their environment. But in males, they are also used for sperm transfer. And here's an up-close view of the male's pedipalps. And look at how highly convoluted, and it's got twists and turns and flips. These pedipalps have a very specific 3-dimensional structure, and that structure needs to fit into the female's reproductive tract when they mate. This is a dissected female reproductive tract. This looks like a black widow male and black widow female reproductive tract. Notice the female reproductive tract has loops, and at the end of that, that's where she's receiving the sperm from the male. So his pedipalp has to fit perfectly with her reproductive tract in order to successfully transfer sperm. And you can tell different species apart by looking at the structure of their reproductive organs. And a different species will have different shapes of these reproductive organs. And I think of it kind of like a protein or an enzyme and a substrate, right? It's like a lock and key fit. They have to fit together just right in order for fertilization to take place. We also have gametic isolation. And this occurs often with species who do not physically contact each other to mate. Examples are sea urchins. You can just look at these sea urchins, and it's not hard to imagine why getting really close to each other is not going to be a very comfortable situation. They've got all those spines protecting them. So when sea urchins reproduce, they just release gametes into the water around them. They will release eggs or sperm. So females release eggs. The eggs go into the water. They're called broadcast spawners, and the males release sperm. And so out in the water around these urchins, there will be sperm and egg. But there could potentially be sperm and egg of different species. And the sperm and egg of these different species are not going to come together because they don't have the correct proteins on their surface to recognize each other. So a sperm, when it comes into contact with an egg, it needs to have a protein that the egg can recognize, and they can bind together, and then the sperm can transfer its genetic material. So if there are different gametes of different species, they will not bind and fertilization will not take place. So these are our prezygotic barriers. Let's talk about some potential post-zygotic barriers. This is when fertilization actually does occur between two different species, but then something breaks down after that. And one thing that can break down is just survival or health of the offspring. And if this is the case, we call this hybrid inviability. And if we break this down, a hybrid is the offspring of parents of two different species. So if you have a frog from a certain species, we'll just call it species A, and it mates with a frog from another species, and this can often happen with frogs, they might be able to have fertilization take place, the eggs are laid, the eggs hatch, offspring are even produced, but those offspring are not healthy, they don't fit in well into the environment, they're not suited to any particular niche, they might have all kinds of health problems, and they die. So they're not the same species, right? They can't produce viable offspring together. That is called hybrid inviability. Hybrid sterility, our second post-zygotic barrier, it's different because in this case, sometimes very strong and healthy offspring can be produced by parents from two different species, but those offspring are not able to themselves reproduce at all. So this is what is happening with a mule. I don't know if you're familiar with this, but mules are actually hybrid offspring, a cross between a female horse and a male donkey. So a female horse bred with a male donkey can produce a mule. And mules can be useful on farms because they're big, they're strong, and they have a certain mix of physical and also sometimes behavioral traits that might be useful for having on a farm. These, however, are sterile individuals. Mules cannot reproduce. They can't produce with horses, they can't reproduce with donkeys, and they can't reproduce with other mules. So a mule is not a species, and horses and donkeys are still two separate species. You can also have the other type of cross, male horse, female donkey, and I think that's called a hinny. It's a different type of hybrid that's produced. You probably are familiar with some of these hybrids, like, for example, a liger, which is a hybrid cross between a male lion and a female tiger, and a tigon, a hybrid cross between a male tiger and a female lion. And you may notice from this liger here that, by the way, these are not crosses that will ever naturally occur anywhere in nature because tigers live in Asia, lions live in Africa. They're very distinct from each other geographically. This is only going to happen essentially if people are manipulating the breeding of these animals, bringing them into captivity and putting them with another species and not enabling them to breed with their own. And often these hybrids that are produced are very unhealthy, could have a number of health issues. One thing that is well known about ligers is they can grow to really large sizes because they don't have the natural control mechanisms that are built into the lion and tiger species themselves because of the different contributions from males and females. So it just works out that way with this hybrid cross, and that can lead to a number of health issues. So this brings us to, we've thought about different ways that species are kept separate reproductively, but this brings us to the question of how do new species evolve? And the evolution of new species is called speciation, and it basically works like this. You start out with a single species, but something happens that over time drives the divergence of different populations of the species. So one group within the species might stay the same and keep the features of the original species, but another group or population within the species evolves differently and diverges enough, becomes different enough from the original that they can't interbreed anymore. And now they are two distinct species, and there has been the evolution of this new species. So what can lead to this type of reproductive isolation and speciation occurring to produce new species? The primary mechanism for speciation is what is called allopatric speciation. And you might need to make a special note of this to write this down. You've got this word here, allopatric. Allopatric means that they are isolated from each other, that they do not overlap, that they don't share a same space regionally, that they are physically, geographically isolated from each other. So allopatric speciation requires geographic isolation of populations for a certain amount of time. Let's see how this might play out. This is a view of Death Valley, and some of you may have been to Death Valley, and if you haven't been to Death Valley and you live in California, I strongly encourage you to go there. It is a really interesting place geologically and geographically and also biologically. It's really interesting to think about this big valley because 50,000 years ago, this whole area of Death Valley used to be filled in with a really big lake. So there was a big lake filling in this valley, and that was in the wet times that occurred before everything dried up about 10,000 years ago. So think about 50,000 years ago, big lake in this area that is now Death Valley, and then about 10,000 years ago, everything starts drying up and this lake dries out, but now there are isolated pools in various locations in Death Valley. So you can see an example of one of these little pools right here, but from this pool to the next nearest pool, there's a lot of land in between. So now these pools are isolated. 50,000 years ago, there was one big population and species of a little fish that's called a desert pupfish. It lived in that great big lake. As the valley dried up and these pools became isolated from each other, these little populations of pupfish got isolated in these individual separate pools. So now you have to think about this population that is cut off from all the other individuals of its species. And imagine that there are 10 or 12 other little pools out here in the valley too, each with its own isolated population. That's the first step. The second step here is that those populations that are isolated evolve differently. Now, I want you to think about this. What are the mechanisms through which populations evolve? And how could that influence the evolution of these isolated populations of pupfish? Could it be through genetic drift? Have you thought about the fact that what was once a really big population has now been isolated into small pools and much smaller populations, right? So there could have been a bottleneck. There could be further genetic drift that leads to the random loss of alleles in populations. Could it be through natural selection? Yeah, right? What about different selective pressures in these pools? What if one pool is saltier than another pool? And we know that some of them are. What if one pool has predators in it and another pool doesn't have predators in it? Then we might expect these populations to evolve differently through natural selection. What about mutation? Is it possible that a mutation could arise in one of these pools and that mutation doesn't arise in any of the other populations? Yes, certainly, right? So there are several different ways through which these populations can evolve differently, and they're isolated from each other. And if that happens over a period of enough time, then that could lead to enough changes in the genetics of the population overall that these changes are enough to lead to reproductive barriers. Even if these fish were to come back together, they can no longer interbreed with each other. Now, I had mentioned the horse and the donkey earlier. I forgot to say that the reason those mules are sterile is because of a chromosomal number mismatch between horses and donkeys. They don't have the same number of chromosomes. So any mutation that occurs that could change the chromosome number, any natural selection that occurs that could lead to different color patterns, different mating behaviors, for example, these could all lead to reproductive barriers taking place. And that's the third step to allopatric speciation, is that the changes that have occurred due to evolution in different situations after geographic isolation, those changes have to lead to reproductive barriers between the species, and that's when you have two different species. They're no longer the same species in different populations. Here's another example of allopatric speciation. This is a local example of the evolution of the Channel Island fox. We're not really sure how long ago it was that some little foxes from the mainland in probably around down near Ventura made their way out to Santa Cruz Island and the northern Channel Islands. But some foxes did get out there. Maybe they got washed away in a big storm event down a river into the Santa Barbara Channel, and then they were able to swim across the rest of the way to these isolated islands. But once they became isolated from the mainland gray fox population, then the population of foxes that were isolated out on these islands, they began to evolve differently. And one way that they evolved differently was through selection for smaller body size. On the island, there aren't as many plentiful resources, and this is a common thing that populations of species that get isolated on islands evolve to have a smaller body size. And that happened with these foxes, and now they are a different species through allopatric speciation. If you visit the Channel Islands, and here's another place you should definitely go to if you're in California or plan to come to California, Santa Cruz Island. You can go out, take a day trip from Ventura Harbor, and see the Channel Island foxes out there. They're all over the little campgrounds, and they're tiny. They're smaller than a large house cat. They're smaller than my cat, for example. Significantly smaller than the gray foxes that live on the mainland. And they're a different species because of this allopatric speciation that started with the geographic isolation of being out on an island separate from the mainland population. Allopatric speciation often happens on islands. It doesn't have to happen on an island or in isolated pools of water. This graph kind of shows you the difference between allopatric and sympatric speciation, which we haven't talked about yet. But you can see here with this allopatric speciation example, you have this population of trees, and a river cuts a canyon. And now we have this canyon cut, and the climate or other conditions might be different on these different sides of the canyon. So if they are, they could lead to evolution and divergence in these populations that are now split by a geographic barrier, and that could lead to allopatric speciation. We see this with the Grand Canyon. There are squirrels, for example, that were populations of squirrels that were isolated on different sides of the canyon that evolved into different species. You could see how this could happen with mountain ranges that rise up and separate populations of the same species. And if they can't get over and intermix anymore, then they could become different species through allopatric speciation. So the next type of speciation, the second kind, is called sympatric speciation. And sympatric speciation does not require geographic isolation. This is when speciation can occur with populations that are coexisting in the same area, but due to some environmental pressure, they start diverging into two genetically distinct populations, which ultimately leads to reproductive isolation and speciation. So let's look at an example of sympatric speciation. And this is an example that is actually currently underway. And I think it's an example that's kind of easy to relate to because we can think about flies and fruit trees. And again, sympatric speciation. Sympatry means geographic overlap. They are in the same geographic area. And this example is hawthorn maggot flies and apple maggot flies in North America. And this is what these maggot flies look like up here on the right. The female flies will lay their eggs in fruit. And usually they'll lay their eggs in rotting fruit. And then the maggots will hatch out within that fruit and use that fruit as a resource. So They specialize on certain types of fruit trees, and they use those fruit trees as their hosts, their parasitic flies. 400 years ago, or over 400 years ago, if you had come to North America, there were no apple trees in North America at that time. Apple trees were brought over later to North America. So before the introduction of apple trees, there were hawthorn trees, and there was a species of fly called hawthorn maggot flies that used the hawthorn trees as their hosts. They are the hawthorn maggot flies. But then apple trees were introduced, and apple trees can grow in the same area as hawthorn trees. And some of those hawthorn maggot flies started using apple trees as host plants. So we have a group of these hawthorn maggot flies that now have done a host switch and are using apple trees and laying their eggs in the fruits of apple trees. And, we're not really sure why that happened. It could be because of competition for resources. These flies realized or just had a behavior that would enable them to use this new resource that wasn't being used by other flies. But the flies that lay their eggs on apples, they have offspring that are more likely to use apples as a host. And it kind of perpetuates the use of that host by that group. So that became kind of a separate population of these flies, and they have been continuously diverging genetically. A part of it has to do with the timing of the fruiting of these different species, because they don't fruit at exactly the same time. So now we would have this group of maggot flies that's using a different host that is fruiting at a different time, and they're going to become reproductively isolated and separate species that are specializing on a different host plant. So right now it's still in the diverging stages. Speciation takes a long time. The very quickest speciation events take a few thousand years, and some take much longer than that. So usually it takes a lot of time. But sympatric speciation is another way that speciation can occur. The by far most common way that speciation occurs is through allopatric speciation, through genetic isolation. That's it for speciation, folks. I will see you next time.